Hello, everyone. A joy to see you all here tonight, attending this live lecture. My name is Dr. Maggie Fostier, and I'm Head of Sustainability in the Faculty of Biology, Medicine, and Health. And today I am hosting the lecture, and it's a joy to be there. So we are hosting the Irene Manton lecture, and this is in collaboration with the Linnean Society of London. The talk today is delivered by a colleague of mine, Amanda Bamford, and is about the Essex orchids, from Essex orchids to tropical frogs, studying and conserving nature. Robbie will talk a bit more, but she's a dear friend and has been a mentor of mine, so I'm so pleased to have her here tonight. Tonight is extra special because it's Amanda, obviously, but also because it's the 10th anniversary of this lecture, which I think you set up initially. And also, you may know this, we are in the bicentenary of the University of Manchester, so there's quite a lot of things to celebrate this year, and this lecture is part of it. 200 years of making a difference, 200 years of learning, innovation, and research, and 200 years of work with the community and incredible work, with the people working here and around us. And obviously, hopefully 200 years to go to forge more collaboration and approaches there. I'm going to give you some housekeeping things. So we are in the museum now. There's no fire alarm planned. So if there is an alarm, we need to make our way outside. There's something serious going on. We are recording the lecture. It's not live. It will be edited after. But as a result, please do not take any photographs. They will be taken during the lecture, and we can provide them after. And if you do not want your photo to be published, please let us know so that we edit you out. There will be a question and answer session at the end. We will be able to take questions live like this, but if you feel a bit shy, perhaps, or your questions are coming to you as we are talking, we have a slido.com code here where you can type your questions as they come to you, and we can go through them at the end. Without further ado, I'm going to introduce Robbie Blackhall-Miles, who is the Linnean Society former vice president, and is a conservationist by trade.
Hello. I'm not going to talk anything about myself. I'm going to talk about that lady in the middle there. That is Irene Manton. Does anybody know what the Linnean Society is? We are the world's oldest society for the study of natural history. People like Alfred Russel Wallace and Charles Darwin, notice I gave him first, the co-orators of the theories of evolution by means of natural selection, presented their theories to the Linnean Society. Robert Brown presented his work on microscopy to the Linnean Society, and through the centuries, the Linnean Society has played host to some of the most prestigious scientists in the world. We are the society that awards the Linnean Medal. The Linnean Medal is basically the world's foremost medal for the study of a natural science. And our current president is the lady right over there on your left, Anjali Goswami. She's going to be with us until May, and then her presidency comes to an end, and she is a paleontologist. Our vision as a society is a world where nature is understood, valued, and protected. And it's very important that those three things come together. So understood through science, through research, valued, that we utilize that research to understand how important nature is to us, and to utilize that value and understanding to then go on to protect it. But who was Irene Manton. Why have we got this lecture happening here today? Well, Irene Manton was a fellow of the Royal Society and she was a fellow of the Linnean Society. She was our first female president. Bearing in mind that the first female fellows were only admitted to the Linnean Society in 1904, she was president of the Linnean Society from 1973 to 1976. So it took a heck of a long time for this learned society to go from having its first female fellows to having its first female president. And then it took a heck of a long time for us to get our next female president, and that was Diane Edwards, Professor Diane Edwards of Cardiff University. And she was the person that gave the very first Irene Manton lecture. Diane pushed for our next female president, Sandy Knapp, and then in quick succession we got Angeli. It's interesting that Irene, Diane, and Sandy were all botanists, and botany was a study that was okay for women. So, you know, Angeli, however, is a very different case indeed, and she studies the evolution of mammalian skulls. Irene has lots of accolades, but I just want to talk about two of them. One is this fern, Polypodium mantonii. It's a hybrid between Polypodium vulgare and Polypodium interjectum. And you have to use the techniques that Irene developed in order to be able to ascertain whether it's a Polypodium cross mantonii. So it's her microscopy techniques that we use to understand the fern hybrid that is named for her. Irene and her sister, Sydney, and it's very interesting that we have someone that worked with plants and someone that works with animals here today giving the lecture, because Irene was a botanist and Sydney was a zoologist. They have a crater on Venus named for them.
Yes.
And it's very specifically because men are from Mars and women are from Venus. And these two ladies, both fellows of the Royal Society, were real kind of at the forefront of women in the natural sciences. And that's why this lecture is really to celebrate women in the natural sciences. And it's the reason why these two ladies have a crater on Venus named for them. But for me, personally, the reason why Irene Manton is so special is because she was also interested in archaeology. And she had this amazing collection of archaeological items, normally telling the story of plants and people. And I use some of the work that Irene Manton did in my day-to-day life, understanding how plants and people come together in the same space. A lot of Irene's collection helped us to understand the history of viniculture. That's the study of the growing of grapevines through history.
Oh.
And stolen straight from Twitter. And we're going to see this picture again, apparently. Similar, maybe. That idea of working with crops and understanding of crops helps us to understand plants. It helps us to understand plants and people. Crops very often help us to understand the natural environment in a lot of different ways. And it brings us very nicely to our first speaker today, Amanda Bamford, someone that I've known for quite a long time, originally studied the impacts of air pollution and global climate change on crops. And And so there's definite links there between what Irene was doing and what Amanda started out doing. Amanda is Emeritus Professor of Plant Sciences here at the University of Manchester. Her teaching interests include, as I say, pollution and climate change. She's held roles at the, as the university lead for sustainability, and she's the associate dean for social responsibility. And Amanda was the person that created this event, that partnership between the Linnean Society and Manchester University. So it's particularly poignant that Amanda's giving our 10th Irene Manton lecture. So I'm absolutely delighted to welcome you, Amanda, to talk about your journey to where you are today. So, Amanda Bamford.
All right. Where do I start? I was dreading this lecture. You think after all the years I've been lecturing that I'd know what I was doing. But this isn't a normal lecture. Normal lectures for me and Giles and other people I know I see in the audience is giving talks on photosynthetic abilities of carnivorous plants, or looking at mitochondrial biochemistry, really sort of dry academic lectures. But the brief for this lecture that the Linnean Society wanted me to do was really talk about my life scientific, any Radio 4 listeners here, to talk about my journey of how I got where I was and my passion and my interests, and really show you, hopefully, by the end of this lecture, that by the end of this lecture, that working in conservation and connecting with nature can be a joyous thing. Very often when you hear about conservation, it's all doom and gloom and going extinct and what we're going to do. And I want to show you, actually, that one, life can throw up surprises, your career can go in lots of different directions, you could be interested in lots of different things, and still, by the end of it, look back and think, that was a really good thing I did then. Or I really enjoyed that, wasn't it amazing? Having that wow factor. And again, my whole life has been about giving that wow factor to other people. So, title of my talk, From Essex Orchids to Tropical Frogs. I don't know whether my Essex accent is still noticeable. Yeah? Well, for my family, they think I took posh. Because a lot of my family are still in Essex. I went onto the dark side and went northward. What for gap? I married a northerner, so that's my excuse. But what am I going to be talking about is how studying and conserving nature and how I've been doing it since the 70s. And I've also, as Robbie said, I'm going to be joined by Matthew O'Donnell, who I've done a lot of collaborative work with. And I'm not going to say too much because I'll give the game away. But again, I'm a plant scientist, but I've never worked alone. I'm interested in plants, I'm a bird watcher, tropical frogs, Rafflesia plants, invertebrates, hummingbirds. I'm interested in everything. So you don't have to be the world's expert on spiders. You can be interested in lots of different things. So for people you don't know, there's a map. There's Essex down here, right down south. So that's Essex. And I grew up in this borough, Thurrock Borough here. And the little dot there is Grays. And that's where I grew up, right on the Thames here. And anybody watch EastEnders anymore? No, do you remember the beginning of EastEnders where they do this zoom and at the end you see the O2 Arena at the... We're about 20 minutes from the O2 Arena. So that gives you a context of where we are. And I know other people here are from Essex, hi Giles, but he comes from the nice part of Essex, the rural fields, and I come from down here on the Thames estuary, growing up surrounded by cement works, power stations. I'm giving away my age here, aren't I? I was a teenager here. There were lots of quarries and gravel pits in the area, housing estates. Get the picture? Thames estuary, industrial. But I loved nature. So why, what would I do living in an area like this? Well, going back to the 1920s, I wasn't around in the 1920s, the Grace Chalk Quarry, which is going to be my main focus at the start of this talk, looked like this. And this is literally a walk from my house. It was abandoned in the 1920s. All right, they made a right mess of it. And other pits, gravel pits in the area carried on to the 1970s. But Grace Chalk Quarry was abandoned in the 1920s, and that's a really important thing to remember. Because by the 1980s, all through that time, 60 years later, Grace Chalk Pit was rewilded. It was rewilded by nature. We didn't have anything to do. It was abandoned. Nobody was interested. No, there wasn't people going out looking for plants and bird watching in my area. Nobody did it. Nobody at my school did it. So this was forgotten. There were still parts of the quarry that looked like this, but there was other parts that had been rewilded. Wooded areas, bit of chalk still left. Some areas of ponds where they'd got when they were graveling, getting the chalk out, they'd gone below the water table, so it was ponds. So it was a great place to live near, and if you live near nature, this was my nearest nature, this chalk quarry. However, if you went back in the 8 years later, that would be your view. And during this time, in the 70s and 80s, I was part of a group that we campaigned to save Grace Chalk Quarry. And when you look at this, you think, well, that's real sad, isn't it, that it now looked like this. But this area, when the 70s, was full of orchids. This is where my love of plants came from. I used to walk down the road, go to a roundabout, climb down the side of a cliff. I don't think I was supposed to. It wasn't open to the public. It's a bit dangerous. But over 7 species of orchids were found that we saw in this quarry. So it was an island of wildlife surrounded by this industrial landscape. And for the biologists here, it really epitomised this successional idea of bare chalk through to open chalk grasslands, shrubs, woodland areas, a lovely mixed habitat. I joined, when I was 11, Thurrock Natural History Society. But I was even more interested in wildlife before then. And would you believe I've still got this little book that my mum gave me when I was 9. And I've still got it, the Observer Book of Wild Animals. Not that there was many wild animals down here. But I loved bird watching. I look back and I think, oh my God, I must have been a really strange 16-year-old going out bird watching. All my friends were going up into London, to Petticoat Lane, to the market on a Sunday morning. I was out bird watching in Grace Chalk Quarry. This society changed my life, really did. I mean, I'm not exaggerating. They met every other Sunday. They went to different places. I was the only young person in the organisation, in this society. They used to pick me up from my house and take me out bird watching. And they took me down to Grace Chalk Quarry one day to see the orchids. And that was it, I was sold. Yeah. So when you talk about organisations that, change the course of your life, this was one of them for me. And Grace Chalk Quarry, well, orchids. Who doesn't love orchids? Eh? Common spotted orchids, see them all over, still beautiful. Pyramidal orchids. And these two hybridise, or as Robbie said to me earlier on, they're quite promiscuous. So they're really, and Ollie at the back will help me out with this. I always go to him, he's our orchid expert. Hi, Ollie. When you try to identify these orchids, because they can be bright pink, pale pink, and we have some of these common spotted orchids on campus. I'll tell you where if you come up afterwards, or Ollie will tell you. But in Grace Chalk Quarry, you also had this amazing orchid. It's called a man orchid, very rare. And look at the flowers, and they look like a little man, little legs, little arms, little head. I mean, orchids, who doesn't love them? And then this one, bird's nest orchid, another rare orchid found in Grace Chalk Quarry. An abandoned chalk quarry, a brownfield site covered in orchids. This is a weird one. I mean, my husband, the physicist, said, well, that's not a plant, that's a fungus or something. And I went, no, look at the flowers. This is what used to be called a parasitic orchid, but they don't really use that now because, in fact, what it's doing is there's all these orchids only grow where there's mycorrhiza, fungi in the soil. Some of them are very, very fussy, like the bird's nest orchid. So you can't pick up an orchid and just pull it up, although people do, don't they? Smuggle orchids and plant it in your garden, it wouldn't grow. If you don't have that fungi in your soil, it wouldn't grow. So this bird's nest orchid, and they've only just found this out recently. They didn't know this when I was growing up and going down Grace Chalk Quarry. What it does is this mycorrhiza makes an attachment to this orchid, and the other end of the mycorrhiza is with beech trees and birch trees and their roots. And there's a transfer of nutrients through this fungi from tree to orchid. Weird, eh? Who doesn't love orchids? Let me test people. What orchid do we eat? Plant scientists amongst you are not allowed to answer. Anybody know? Have it in a kitchen cabinet? Yes, it's the only orchid we eat. And the other thing about orchids, their seeds are like dust. Really, really fine dust. Another reason why they're very difficult, almost impossible to propagate and grow. It's like dust. So if you ever next time you have really good quality vanilla ice cream, have a look in and sometimes you see little black dots in the ice cream, and that's orchid seeds. It has to be good quality vanilla ice cream. I could go and give a whole lecture on orchids. Aren't you surprised, yeah? But I won't, but I just wanted to tell you about what inspired me. So if you went back to Gray's orchid now, right, this is the map. So this is the extent of the pit when I was growing up, all round here. And this is that housing estate I showed you the photo of. And this area here, the north of the site, is now a nature reserve. So we lost the fight and couldn't save all the orchids here, even though they tried to persuade us they're going to move them north, but we used to tell them you can't do that because of the mycorrhiza. So it's a half success story, that there is still some of that site left. And this is what it looks like now. If you went there now, this is what you'll see. And the problem is that now they had to have heavy management to try and go and cut down a lot of this woodland, a lot of these shrubs, to enable the orchids to stay there. Because the orchids, a lot of them like open areas, open grasslands, except the bird's nest, it likes trees. So now they're having to do a lot of management, Essex Wildlife Trust, going back and cutting down and back a lot of this to keep the mixed habitat. Because like classic biology, eventually if you left it, it will turn into a woodland. So when we're talking about wilding and saving nature and restoring nature, one of my problems when I hear people talk about this, I want to know, well, saving it to what? Do we go back and make it all bare chalk again? Do we go and cut down all the woods so we can get more orchids? But then there's rare red-listed beetles and insects really down in Grace Chalk Quarry. So do we make it so it's a habitat for them? So that's the big quandary with wilding and rewilding for me. What do you want it to look like? How much management should we put in there to keep it how we want to look like? We do it for orchids, but would we do it for a little robber fly that's on the red list species that's found in Gray's Chalk Quarry? So I was fascinated. I wanted to learn more. I wanted to go where people look like me and were interested in things like me. I wasn't going to find that. I'm sorry anyone's here going to listen to this from Gray's. They weren't going to get it there. There was only me in my whole school, and there's about 1,000 people there that went bird watching, that went out planting, but looking at plants. So I was the first one in my family to go to university. My parents never went, but luckily they encouraged me to go. They could see my passion and my interest. I wanted to learn more. I wanted to find out more about orchids. I wanted to find more about nature. So I went to university. And part of my degree there, I had 2 placements. One, I went to Huge Seed Bank, and I actually saw orchid seeds for this first time. And I saw conservation in action, about how conservation doesn't have to be going back and planting trees. It could be preserving the seeds for future generations or replanting the seeds. But for orchids, only now recently they're still working on how you can grow orchids. And they've actually they've gone back to Gray's chalk quarry, literally this year, collected the mycorrhiza and managing to grow these orchids in the glass house. I also went 6 months to Silwood Park, which is the field station for UCL, and there I realised I wasn't the only one. During lunch hour, we went out with botanists, entomologists with their big nets, bird watchers, fungi people. I used to open the fridge and look in there, and there'll be a bracket fungus in there for tea, you know. And everyone thought it was perfectly normal. I met my husband there as well. Silwood Park changed my life as well, because I realised that there was lots of other interests, not just botany and bird watching. There was all this other wildlife that I could be interested in. And for the plant people amongst you, one of the people I was really friendly with, someone called Mick Crawley, who's really famous, and he used to come out on the walks with us. So obviously, you know, passing on the baton. And certain things stick with you. I'm an image person, and I remember going out on field courses as an undergraduate, and I still can close my eyes and remember this field we came across near Bath, full of snakehead fritillaries, which are rare plants. And it worried me. I thought, why? They're supposed to be rare, but locally abundant. So if you go in the right place, and you can imagine this whole field full of these beautiful flowers just sort of fluttering in the breeze. We just stood there for ages just watching them. I got to the end of my degree. I thought, this is great. I found the place I belong. I want to find out more. So I went to the University of Lancaster to do my PhD and do a postdoc. And you remember back, I showed you that photo of the cement works and hanging out the washing on the wrong day or breathing all that cement in, and there was a power station and Procter & Gamble chemical works, so you could smell the chemicals coming off it. So I think I always, the back of my mind was thinking, well, that's what's happening to us, what's happening to the plants. So I went to Lancaster and we used equipment like this to expose trees and crop plants to pollutions like ozone, which is a really phytotoxic air pollutant. And we were looking at what level the plants could tolerate, because at that time they were still working on the standards for air pollution standards for regulations. So people wanted to know, well, what level should we set for ozone, for example? So that's the research I was doing there. And then Gary and I decided we were a bit fed up with the rain in Lancaster, and we decided to go somewhere a bit sunnier. So I got a postdoc in the University of Florida, and we're now talking about late 80s, early 90s, where this thing called climate change was being mentioned. And it wasn't a really big thing at that time. It was still in the realms of the scientist and research, and it was just starting to be talked about. So I went there and joined the agronomy department at the University of Florida, where I worked on the impact of climate change on rice, which is one of the major food crops of the world. So it's really, I felt it was a really, really important work to be involved in. Gary joined me out there, and he got a job working for NASA, working supernovas. I think maybe he might win that as being the coolest. But I still think it was a really good place to do this research. A huge team. It was very expensive research, big chambers full of growing rice and all different temperatures and different concentrations. So it was very expensive research, big lot of resources. And now while we were out there, we had our first. Oh, I forgot something for the students here. Protesting. I belong to a society called Eco Action. And we went out, this is a CND march, but I went on Save the Whale marches in London with huge inflatable whales and things. So it was always an undercurrent of action as well. It wasn't just academic research. All the way through, I was interested in making a difference somewhere, in some way. So I got back to Manchester. By this time, I had 3 boys under 5. Research was very difficult to do in those situations. In those days, there really wasn't part-time research roles. So I really wasn't involved in a university at that point. But then a friend of mine who was a professor here, Professor Mike Eames, rang me up and said, we just had somebody leave without much notice. Could you come and take over their lectures? So I went, well, all right then, but I do have 3 little boys. He said, no worries, you come in, give the lectures and go home again. I thought, well, all right then, that'll at least get me back in the field, getting back into the world of work. And I loved it. I regretted leaving my research behind, but I really felt I could make a difference lecturing, taking students out into the field, freshwater ecology. This is a course that Keith White and I used to run in Greece, where we used to go to the lakes and rivers, monitoring them for pollution. You'll start seeing a theme here. This is Giles, who I know is in the audience. We used to run a field course for master students to a chromium-contaminated site. And this is us in the rain, identifying grasses in the rain. This is Keith and I. We took master students to Paris Mountain, a copper mine with acid mine drainage. He talked to them about the impacts on freshwater. I talked to them about the impacts on the plants in this area. Cecilia Medipin and I went to Myanmar and ran a series of workshops for university lecturers in Myanmar so that they could then teach their students about how to go and do lake and river pollution monitoring. We even, this is Cecilia here, ran programs for local communities so that they could go and monitor and look at their own freshwater systems near where they live. And Andrew Dean, who's now at MMU, and I can't believe I did this, actually ran a program for school children. So Natalie I don't know whether Natalie was around then, but I can't believe we took them out. We gave them waders and meters and said, there you go, let's go and measure the pH and look at the invertebrates. And we didn't lose any students. None of them fell in. We brought them back to the classroom dry, but they loved it. Can you imagine? Some of them we brought into our labs at Manchester University so they can use our beautiful microscopes. £2,000 microscopes we gave to these high school kids to look at what they've collected in the rivers. So I love this. Raising skills and awareness and getting that wow factor became my life, really. And then in 2014, the Faculty of Biology, Medicine and Health said, well, you're doing all these field courses, could you run a tropical one for us? Of course. And I thought, where am I going to do this? And I had a friend who worked here at Manchester Museum, he only retired last year, called Andrew Gray. And I knew he'd worked for many years in Costa Rica. Now, Costa Rica, in case you don't know, is over here. This little tiny isthmus between North and South America. And it's in the area, the Mesoamerican biodiversity hotspot. So there's, in this tiny little country, the biodiversity is out of this world. And also, Costa Rica is one of the leading countries for sustainability and conservation. Over 30% of Costa Rica is under some sort of protection, either national reserve, a local reserve. Over 25% of the country. So I thought this would be a fabulous place to take students to. And I wasn't wrong, was I? And we're still running this field course today, and Matthew has been on this field course as well. We take Dmitry Luganov, who's a world's expert on spiders. Andrew's coming, an expert on frogs and reptiles. I go along for the plants and also my bird watching skills for hummingbirds. And we do moth trapping, we do bat detecting, you name it. It's the whole thing. And what I want to do is show you now is a film to give you a bit of a flavour of what it might be like, what it is like going out into the Costa Rican forest at night with the students on our field course. And also, what we did this time, we interviewed the students just as they came out of the forest to see what impact it had had on them. Let's see if this works. So it starts getting dark, and I tell you, at night it gets really dark in the jungle. So we put our head torches on, and then all the sounds start coming out, and we see frogs, spiders, snakes.
So I'm here in the Costa Rican Amphibian Research Centre. I've just had my first experience trekking through the rainforest. Really, really good experience. I've read about kind of a lot of species we came across prior to coming here, and it was just so much better to be able to see them in the natural habitat.
I'm not great with like dark and creepy crawlies, but you're in such good hands, and like when you see the frogs, you just forget everything else because like the colours are spectacular. I can't even describe how amazing it was to see them in the wild.
That's it. I've got a longer film, but I thought I'd better not. But if you want to see these frogs, you can see them in the vivarium here in Manchester. How cool is that? Yeah? You don't have to go to Costa Rica. So because of our work in Costa Rica and the field course, I got involved in some very specific projects around endangered species that are found in Costa Rica and Panama, and I want to tell you a bit about that. So can you see what I've done? Gone from orchids to tropical frogs. Yeah, it's weird how things work out. So I'm going to talk to you a bit about our project on the Lima leaf frog. I'll leave Matt to give you the Latin names. The harlequin frog, or the Atelopus, and also, going back to my bird watching passion, great green macaws. So this is, again, I could give a whole lecture on each one of these slides, but I won't. So this project is all about involving people. So it's ecological habitat restoration. You go out and there's putting out, they like laying their eggs in holes in trees. So they went out and put buckets out for them. Very simple. Nothing technical there. In situ and ex situ conservation, so conservation in the area where they're found, but also we've got a population here in Manchester. Local schools, and this is where I came into the project, we set up a whole programme around environmental education, because a lot of the schools there, they didn't even know they had this very rare endangered frog right next to them, a bit like me with the orchids. And so we set up, you can go on a website and there's videos on there for the schools in Spanish and English, all about climate change and the impact on frogs and about the Lima leaf frog. And we had printed and we sent out thousands of these booklets for the schools to use, all about frogs and also about conservation. And again, this will come up again and again, and Matt will talk about this. We didn't do it alone. It's all collaborative. One person is not going to, I don't think, change the world, to be honest. Most of us are not. It's all about everybody coming together and using their skills and expertise. So it's a collaborative thing. And also it's important, this is a little boy that we went into the school and we took in loads of T-shirts for them. It's all about raising awareness, giving them the skills, involving the local communities as well, just as important. So it's the whole package is really important to us. And then the ambassador to Panama, Andrew Gray, and I were invited to give a talk to all the ambassadors of Latin America, would you believe? Absolutely petrified. Down in Palmau, really posh, you know. We all had to go there, and we took some of the frogs from the vivarium, and we talked about the Lima leaf frog project and our field courses. And afterwards, the ambassador of Panama came up to us and said, well, really, can you do something for us in Panama? there's this group called the Panamanian Wildlife Conservation Charity, and they're working to try and save this endangered harlequin frog. And again, so they were starting up doing population monitoring, but they hadn't had all these other aspects to their project. So we came in and we helped them set up, again, a schools program. Lots of these went into schools. And so what I want to do is show you a video that the Panamanian PWCC, this charity, made for us to show what they were doing. So I didn't go over there. The local scientists, the local community, they do the delivering, and I think that's really important. I can't speak Spanish either. I'm trying to learn, but I'm not very good. So I'm going to show you this video.
Well, we are here in Alto Ortiga. It's a community in the Caribbean side of Santa Fe, and we're having this workshop trying to address the issue of the harlequin frog conservation. We planned this workshop for many months, and finally we arrived here in this school. What we did today was just a presentation about the project, about Santa Fe, about the frog in general terms for the kids, talking about why the frog is important and what happened to the frog in the field, why this frog disappeared, and why it's so important for us as humans to preserve it. And after that, then we deliver a booklet that has been part of this partnership with the University of Manchester to provide information and educational materials to communities here in Panama, especially in Santa Fe, through this Atelopus project. And we play with them. They made some of the tasks that are inside the booklet. They love it, they like it, they repeat the name of the booklet many times, and they keep doing it along the workshop. Now we come with this initiative, which is a partnership between Panama Wildlife Conservation and with the Manchester University. So Manchester University, in my view, provides this expertise, the academic advice to the project, and also with the partnership of PwC that can, come here, provide funds and resources to this project to allow the project, continue in the future. The new generations are the future of this planet, and we want them to be prepared for the challenge that they will face. And we have the responsibility to transfer the knowledge and try to engage them to preserve, really be active in the future to preserve this environment that they have.
Isn't that gorgeous? Yeah, so it's the same ethic. We don't partner up with just anybody. They've got to have the same ethics that we have and the same passion that we have. So I'm going to hand over to Matt because of those two projects, there's been lots of opportunities. We've gained the trust of the local community, the ambassadors, the universities out there, and we've been able to expand into other projects. And also Matt's going to talk to you a bit more about amphibians because that's his expertise. Matt.
Brilliant. Well, thank you very much, Amanda. Tough act to follow, but I'm delighted that you asked me to support this lecture and to talk a little bit about the frogs, the real stars behind the scenes of the vivarium here at Manchester Museum. So one of the reasons why we do have this focus, and I've worked with amphibians now for a number of years at the museum, is amphibians are in danger. A recent report that I was very fortunate to be involved with is the State of the World's Amphibians, the second global amphibian assessment, which was conducted by colleagues from the IUCN and involved expert assessors from all around the world. And we assessed over 8,000 species of amphibian. So no mean undertaking. This is a big report. And what we found in this report, so I contributed to the Costa Rica aspect of it, so only 220 species, so a small proportion of that. But what we found is actually amphibians are the most threatened group of vertebrates on the planet. So over 41% of the over 8,000 species we analysed are threatened with extinction in the wild. So that's really, really stark. So it's more than the reptiles, more than the mammals, more than the birds. It's really, really stark. And one of the reasons for this are diseases, climate change, pollution, habitat loss and alteration. It's manifold. And we often describe this as a toxic cocktail where one individual factor on its own might not be enough to knock these species out, but when you combine them together, you end up with a situation which is intolerable for these species. Amphibians are very sensitive animals, and we often describe them in the herpetological community as the canary in the coal mine, which kind of harks back to that early use of canaries in mining operations where they would take these very sensitive songbirds down into the mines who would be happily chirping away until some gases were released. Well, obviously the miners wouldn't be aware of those gases. They didn't have a smell and they didn't have any of these fancy bits of technology to hand that we have these days, but the canaries would be. So they would stop singing as soon as these gases were released and everyone would be evacuated. And that was an early warning sign that something was really wrong in the environment. Well, amphibians are doing that to us on a global scale. So they're the early warning sign that something is going wrong globally and that we should all take notice. So one particular factor that's causing really profound declines in amphibians is a disease called the chytrid fungus, which forms a disease called chytridiomycosis, which affects the skin of an amphibian. And the skin of an amphibian is very permeable. They often drink through their skin, they breathe through their skin in some cases, they absorb minerals through their skin. Now, if you have a fungus that interacts with that membrane in a harmful way, these animals can very quickly go downhill and die. And unfortunately, we've spread this fungus disease all around the planet and it's caused massive declines. Over 500 species have vanished, have been impacted by this fungal pathogen, which makes it the most, what was the exact wording? The greatest loss attributed to a panzootic that we've ever seen. So this is like a pandemic in the animal kingdom. So we're losing species, potentially up to 90 species have gone extinct because of this fungus. So that just kind of gives you an idea of what we're dealing with. So building on the amazing foundations of the work that Amanda and Andrew did here at the University of Manchester, we've been able to establish some really amazing collaborations out in places like Costa Rica. And the reason why we focus on these areas is because of the biodiversity. There's lots of species there, but also because these are often the areas where the impacts of these diseases and things like climate change are being felt most extreme. So one species that we work with is Incilius holdridgei, the Holdridge's toad, which is, I think, a very beautiful toad, but I might be alone in that. And this species was thought to be extinct for around 30 years. It was declared extinct. It completely vanished from its historical range, and the main culprit was this particular fungus. And it was actually rediscovered by a student group that were trekking through the forest with their lecturers, very similar to what Amanda does with all of her students on these field courses, and they found these little toads. And they took some pictures and sent them to a good friend of mine, Juan Abarca, who is a resident amphibian expert of Costa Rica, and he was very excited by these pictures of tiny little toads. And he rediscovered the species. So an amazing discovery. I've been to the site, Amanda's been to the site. It's hard to get to. It's really, really remote in the Braulio Carrillo National Park. They're found in one mountain, and this is basically the entire breeding site of this toad in the wild. It's a puddle under a tree. And I am not exaggerating when I say puddle. It's about this big. There's probably less than 50 mature adults in this population, so that makes this arguably one of the rarest species on the planet. Now, unfortunately, when you're trying to save species, you often need data, and that was one of the biggest takeaways from our involvement in the IUCN reports. When they were assessing all these species, we need data. We need to know where they are. We need to know the population dynamics. All of this data we are really, really lacking, especially for groups like amphibians. So one way that we can help with that is through my research and research that was established here at Manchester in a collaboration with the University of Salford, which is using environmental DNA. And this technique will help you to detect tiny little toads like this through their DNA that's left in little puddles like this. Now you might think, well, why would you have to use this technique? Why not just go and look for them? I've been there 3 times, never seen a toad.
I've seen one.
Amanda beat me, she found one. but they're nigh on impossible, I think, to find in the wild. They're fossorial. They live underground for almost the entire year. They only emerge for a couple of weeks for breeding. And that timing used to be really regular, but now with climate change, we're having less rainfall at the same times of year. It's really interacting with the activity of these animals, so it's really difficult to find them. So using techniques like this, which are non-invasive, we just filter the water, put the water back, we're capturing DNA, and we're hopefully finding out exactly the distribution of these animals in the wild. Because if we don't have that data, it's really hard to present a case to help intervene with that species to conserve it. So when you approach governments or different organizations, they often want data, and that's what we're trying to do. And then this is another project I'll quickly talk about, which is focused on this beautiful tree frog. This is Costa Rica's, well, one of Costa Rica's most recently described species, Plalocohyla celeste, which is a mouthful, the Tapir Valley tree frog. And it was described and named after this beautiful river, the Rio Celeste, which is found in Bijagua in Costa Rica, right next to the Tapir Valley Nature Reserve, where this species was actually found. And it is that blue, even more so in real life. It's an amazing area. And we were invited in by our friends and partners at Cladoc Amphibian Conservation. They wanted us to come and support this project because, unfortunately, this is what the site normally looks like here. It's an 8-hectare wetland. This is the only area in the world where you'll find this species of tree frog. It's not yet been designated by the IUCN, but when it is, it will be critically endangered because it is only found in this one wetland. This is what it normally looks like, and this is what it looked like earlier this year. So we're having exacerbated droughts in Costa Rica, and as well as a lot of areas of the planet because of climate change, human-driven climate change. And we're seeing these really pronounced sort of dry periods causing real issues for amphibian populations. If you imagine that's one area in the world where they're found, that's all your eggs in one basket. If you have an environmental disaster or something happens, that population could potentially be lost. So we came in and we're working with members of the local community who are absolutely delighted about this amphibian discovery. They're really proud of it. And we've been helping them with some amphibian surveys that they've been doing in this area of Costa Rica. That information then will be paired with the information we get from environmental DNA, and that will then give us a really good evidence base that it's worked, and we can apply that to other sites where they're trying to discover potential habitats where this animal might live or where it could be introduced. But again, before we can do anything, we need data. So that's one of the reasons why we've been invited into this project. And again, this was all made possible. All made possible because of the amazing sort of collaborations that Amanda developed in Costa Rica. So here's some of the great people that we've been able to work with. This was the IUCN Red List Assessment Group from Costa Rica. So there's a skinny version of myself just up there at the top in 2019. And then we've got amazing partners from the different areas. This was Providencia, where they allowed us to stay in this amazing eco farm. We've stayed and worked with lots and lots of students from the universities in Costa Rica who've been assisting in the research that we're conducting. And then obviously myself and Amanda. I had to sneak a photograph in. He's been really amazing and so supportive of these projects. But one thing I just want to finish with and to really stress before I hand back to Amanda is that all of this conservation is only possible through these collaborations, and that is the way that we've always operated. We're really proud of that work, and we're really proud of the connections that we've developed with our partners. So yeah, Amanda, that's back.
And I'm sure, that's just inspirational, the work that Matt's been doing. He's also doing it as part of his PhD as well, so I'm sure it's going to be carrying on for quite a few more years, hopefully. So I want to finish off going back to bird watching. Although I'm a professor of plant science, I still go bird watching. And PWCC, that Panamanian wildlife charity that I mentioned before with the Atelopus that I'm involved with, also have projects with other endangered species in Panama. And one of them that I've been involved with is around this beautiful bird called the great green macaw. And so we used, again, the practice we learned from the frog work we've been doing. We actually, with my final year students, were involved in their final year project in designing booklets that we, again, sent out to the schools in the area. So you start seeing a theme, you know, once something works, you stick with it. And I was really glad to be involved in this because for me, as a plant scientist and a birder, they were involved, part of this project is the ecological habitat restoration. Now, you all hear this thing about planting trees, don't we? Let's plant trees. But my mantra is it's got to be the right tree in the right place. You can't just go and plant trees, any old tree, and then ignore it and hope for the best, which is a lot of places do. This project is really well thought out. It's called LEAF, and part of their ecological habitat restoration, first of all, we employed local community to go into this place, very remote place called Cerro Hoya National Park in Panama, which I went to, and Andrew and my husband came with us that year. And what they did was they actually monitored the great green macaw population to see how many were there, what they were doing, what they were eating, what trees were they nesting on. And then what they did was, after that monitoring, the community has set up a nursery to grow the trees and plants that were specifically being used by the great green macaw. So it wasn't just planting a tree, it was planting a tree or a plant that the great green macaw either ate or nested in. And then the local community would then be looking after those trees. It's actually on local community land. They donated their land, was right next to this national park where there's this population of great green macaws, which is a very critically endangered bird species. And what you can actually do, if you want to get involved, this LEAF project, you can now go onto their website and you can sponsor a tree. But it's not just a tree. You can choose which species you want to sponsor, and it tells you all about that tree and that the great green macaw eats this tree, or the almendro tree, that the only one tree that the great green macaw nests in. You can sponsor an almendro tree. And they geolocate it. Once it's planted out, they then put it in the nursery. And then once it's planted out, they geolocate it so you can know where your tree's gone. So I love that because it's communities, they're getting an income from it. So it all makes sense to me. And the schools are involved as well. And I forgot to mention, you know, things always come up with the Atelopus project. There's now an Atelopus festival every year in that area. Well, they have a big bit like our Manchester festival that we have, community festival. They have a community festival, and people from all around the area go to have an Atelopus festival. They have dancing and music. So I want someone's phone, not that sort of music. So I want to finish off by showing you another video all about the great green macaw and the project that they're doing there. that generating an income for the local community, I think, is really important, which a lot of conservation projects forget about. That, you know, you need the local community involved, that they will then carry on looking after that area. They have an investment in that area. Part of the Atalopus project, we actually trained local people and paid them to go out and do the monitoring for us for the project. So again, they had an investment in the project. It wasn't just come and look at this beautiful frog. So hopefully you've got some idea of where I've gone from orchids, tropical frogs, field courses, all the way through. It's conservation and nature. So by studying nature, you can then get engaged with nature and hopefully then go on and help preserve nature and also restore it. So going back to my time in Grays Convent High School, it's actually an all-girls school, which at the time it's not as good as you think because it was an all-girls school, there wasn't many physics teachers around and chemistry teachers, so we didn't have a physics and chemistry teacher. I had to learn physics evening classes with adults in a year, and the chemistry was taught by the maths teacher, because he actually was a chemistry teacher, but we didn't have many maths teachers. So going to a all-girls school for me was quite challenging, being, you know, in a STEM person. It really wasn't me. But I've got involved back into the school in terms of this Forest Future First organisation. It's a government organisation, and they approached me and said, could we use you as an example to say, well, even you living in Grays, going to Grays High School, you could end up going to Costa Rica and working with tropical frogs and orchids and things. So yeah, I thought, yeah, that's fine. You can use my picture. And talking about Wallace, I actually found out more recently, looking at some of these images and about my school, because I've not been back to Grays since I was 18. That tells you something. One of the buildings as part of my high school, I found out, it's called the Dell, and it was the house that Alfred Wallace lived in. Isn't that weird? I never knew. Nobody ever talked about it, but now it's actually quite well known, and there's a plaque on this building that's in my school to Alfred Wallace. And the other weird thing is, it's made out of concrete. It's one of the first houses ever built out of concrete. How weird is that? But anyway, I just want to, I just want to thank everyone. And some of them are people that have been really lifelong colleagues here. I really want to thank you that you've supported me, encouraged me. We've gone out in the rain and the snow and inspired me, helped me stay connected to nature, showed me that there's a place where I can belong, find my tribe. You know, I was so lucky to meet these people and have them in my life. Not just here in Manchester, but the people we've worked with in Costa Rica and Panama and Myanmar. You know, it's been an honour. And also my last little words are, it doesn't really matter where you're from. Growing up in a council estate in Grays, surrounded by cement works, or in an urban environment, you know, brownfield sites are very important, because not only can they be biodiverse, but they can be your nature on your doorstep. You don't have to go somewhere special. You don't really have to go to Costa Rica to see wildlife. There's lots on your doorstep. And like me, you can get involved in the wildlife and the nature around you. And we're very lucky in Manchester that we don't have to go very far to find it. And as Giles and I know, he runs a field course called Urban Biodiversity, where they take students out to local sites all around the Greater Manchester area, looking at biodiversity. So that's what you need to remember. So I'm going to hand over to the last slide, so you can let Matt have the last word, which I think is really important. We've shown you what Matt and I have been doing, but we also want to show you where you can get involved in some of the things we've been talking about.
Thank you very much, Amanda. So if you would like to find out more and support some of these projects that we've talked about today, a bit of a plug for our sponsorship of Sponsor a Frog. You can find that on Manchester Museum shop. And that is an amazing way to put funds towards our conservation work in places like Panama and Costa Rica, and all that money gets transferred directly to our partners. So it's a really worthwhile and exciting way to contribute to conservation. And you also get to come behind the scenes and look at our amazing frogs, some of which I've got down here, which I might show a look if anyone wants to have a look at them. And also that's the link for Leaf Wildlife, which is the amazing tree program from Panama Wildlife. You could also walk all the way down the corridor, about 5 minutes, to our vivarium, which is where our amazing amphibians live here at Manchester Museum. And you'll be able to see lots of the species that we've been talking about, as well as lots of other really beautiful frogs. And you could also visit Wild, which really explores some of the themes that we've discussed today around rewilding and how we go about these challenges that we're facing as a society globally. And it's a really great exhibition. It's only quite recently opened, so I would strongly recommend that you also go and check that out. But finally, as a little surprise for Amanda, I have a lovely message from one of our partners.
Everyone, I'd like to take a moment to celebrate someone who has been incredibly important to Panama wildlife conservation, Professor Amanda Bamford. A few years ago, Amanda visited Panama to help with the efforts to protect the remarkable harlequin frogs and to experience the natural beauty of our country. Her dedication and support had been absolutely invaluable, and we are very fortunate to call her not just a colleague, but also a dear friend. Today, we are not only celebrating Amanda's incredible achievements, but also the powerful contributions women like her made every day in the field of the natural science. Thank you, Amanda, for everything you have done. Hello, everyone. I'd like to tell you.
We'll watch it again. There you go. Thank you very much.
Now that we're going to have a Q&A. Anyone got any burning questions? We're going to set up ourselves over here. Right.
Nice, wasn't it? A bit emotional at the end, I think, for everyone. So if you have any questions now, it's time to ask them. We can repeat the questions perhaps for the recording, so don't feel offended if we repeat the questions, and we have to talk for the microphone for the answers for the recording as well. We also perhaps will have some questions from Slido that Sam will pass on to me. Anyone wants to open the question session? Well, if you don't, I will have one. Okay.
We have one.
Oh, sorry. Plenty. Holly first, and then after that.
With all the issues in conservation, even in frog conservation, the question is, how do you as a team set conservation priorities? How do you decide which is going to be the animal on the next pamphlet?
Do you want to repeat the question? So how does one set priorities for a conservation with endangered species? Do we prioritize one species over another? How does that work?
That is a really good question, first of all. And it's a question that as a conservationist I've had to grapple with many times, and there isn't one answer. There's lots of answers. I think it depends on the program that you're working with. So I know the Institute of Zoology have a fantastic program called the EDGE program, which works to identify evolutionary distinct and geographically endemic species. It's basically rare animals from evolutionary
distinct and globally endangered.
There we go. I should have practiced that, but I didn't know. So these are species that really require conservation attention, and if we lost them, we would be losing a significant evolutionary branch, for example. So that's one way you could look at it.
And the other way we've talked all the way through is about the collaboration. That we trust our partners, we trust what they're doing and what their motivation is. So that's another criteria that we have all the way through.
And often these species as well, they might be a flagship species. So the great green macaw, I think, is a fantastic example of that. That by conserving them, by working to restore these species, that it benefits so many other species that inhabit the same habitats.
Yeah, that's very much. Yep, that's on. It's about the best bang for your buck as well. So if you've got a species that is going to then have a knock-on impact for a whole range of other species that need help, then, you know, that umbrella species, that flagship species, then choose that, because you're going to have a really big impact with minimal input, rather than having to put a lot of input into a lot of species you can actually get.
And that's the great green macaw project, is that it's an umbrella species. The almendro tree is used by lots of species, and the great green macaw is the only species that can crack the almendro nuts, and then that can be then used, their discards used by other species as well. So yeah, you can look for umbrella species, a really good one as well. Another question?
Someone wants to shout their other question? Can't have been the same one exactly. There was 2 hands up there. Dr. Hannah?
Well, I didn't think there was any questions, so I was going to ask, are you hiring? Thank you for your thoughts. It's really amazing thing to do. It's just so... Do you feel like you have to get in quite early to get this seems like popular work? People would love to be doing it. How does one start to actually work with it? What's this contribution?
So it's a career question. So how does one become a conservationist early? You know, you started when you were 9, didn't you?
You need two, I think you need 2 things. You need passion and you need to study. Yeah, you need the knowledge as well. That's where you need to start off, you know, so that you know that you're doing good. You're not just like the planting a tree, that example. So you've got to do it from a point of knowledge, so that you know you're doing the right thing. A lot of rewilding, a lot of brownfield sites are considered as wasteland, and they've been built over. But in fact, when you look back, they realise that actually they can be just as diverse as a green space. So knowledge and passion are the 2 things.
Are you going to add something? I was going to... We had a bit of a discussion earlier where clearly some people want to make a career out of this, but we also talked about now there's an opening with some apps and things like that where a bit of citizen science can happen, and we can all become a tiny bit more involved, if not ultra knowledgeable. Do you want to talk about this? The iNaturalist and so on.
So in a few weeks' time, I'm off to South Africa to look for a species of plant called Proteoholoceraceae. Unfortunately, a couple of years ago, most of the population was burnt, and it took it from being just vulnerable on the IUCN Red List to being internationally critically endangered. The way that I'm going to be able to utilize that, find out where that species is actually utilizing an app, iNaturalist. And those records are invaluable, but those records are being uploaded by normal people. Hikers. The last records for Proteoholoceraceae were 3 hikers that went into the mountains of South Africa and happened to find those plants. And so that data is then fed into the global biodiversity information system, and we're then able to utilize those records as scientists to understand where those species are, how many of them there are, etc., etc. And so actually, you can be doing an awful lot to help us with our work, just by being citizen scientists.
If you've not used iNaturalist, it's an app, free app, and it's really easy to do. You just take a photo and upload it, and it will help you identify it as well. So if you want to go and identify a really weird sedge that no one, you don't want to, upload a photo and it will help you ID it. So anyone can do it. We do it on the field courses. We have competitions who can ID the most species. So again, as Robbie said, it's really, really easy, and the data is contributing to real research as well.
This question is about the What factors do you think significantly impacts the global population because of their sensitive skin, but they can also be immune to other pathogens? So why is it these particular pathogens?
Do you want to repeat?
Yes. So why is the chytrid fungus, this fungal pathogen, impacting amphibians globally? What is it about this fungus? So, I mean, there's a multitude of reasons. One of them is that the latest sort of research suggests that this fungus originated in the Korean Peninsula, and yet was spread around the world by human activity, so through the farming of amphibians perhaps, through the use of amphibians in research for early pregnancy testing, believe it or not. So there was amphibians being shipped around the world and potentially releasing these fungal pathogens into the environment. Now, what's important about that is that you're essentially taking a disease that the wildlife of those regions have never experienced before. So just like when we had the COVID pandemic, we were experiencing a novel pathogen. The chytrid fungus was a novel pathogen to all these amphibians. They hadn't evolved in concert with it, so they had no natural immunity. Now, what's really interesting, and part of the research that we're doing here with our collections, is to try and identify why some populations of frogs have survived or rebounded in the presence of this fungus. And I think looking at the immune phenotypes, the ability of these animals to develop immunity or survive in the presence of this disease could really inform how we go about protecting these animals. And we learn from nature and use that knowledge to help protect nature in other areas. So it's really, really exciting. And it isn't all doom and gloom, as Amanda really...
And also, don't they, the Atelopus, some of the populations, they sunbathe, which I think might help get rid of the chytrid fungus on the surface. So it's really, really fascinating. That's why I love frogs as well.
I'm just going to add that it's, the chytrid fungus is just one of a multitude of different pathogens that are being spread around this planet that are affecting our biodiversity. I work a lot with phytophthora, water molds, they're oomycetes, and they're being spread on people's shoes. We've got a phytophthora that's impacting the kauri trees that the kauri trees in New Zealand had never had to deal with before. Phytophthora cinnamomi affecting the fynbos in the biodiversity hotspot of Western Cape of South Africa. We've got phytophthora that are impacting our juniper trees here in the UK and our larch trees and our pine trees here in the UK. Ash dieback, etc., etc. It's a suite of pathogens that have been moved around the planet that are impacting our biodiversity, that our biodiversity has never had to deal with before in the way that it's dealing with it now.
Yeah, and actually, at our field courses, we make sure we all wear wellies because of the snakes and everything else, but also we make sure they're all washed before we leave the sites. Sort of, isn't it? And Matt and Andrew have really made us very aware of not transferring on our Wellingtons. So researchers as well have to be very careful about what they do and where they...
It's giving me a perfect segue to a question on the Slido, actually. When you were coming back to the UK and the endangered species here. Here there is a focus on Manchester. Does Manchester have their own plant or species to protect? And what about, for example, the Manchester poplar? Did I pronounce it well? Poplar?
Yeah, Bobby will do that.
So the black poplar tree is Britain's most threatened timber tree. It's threatened lots of different ways, but but Cheshire and Manchester has been historically a stronghold for the species in the UK. Just a little bit further north, we've got the Lancaster whitebeam, Sorbus lancastrensis. That's also IUCN red listed species and endemic to the UK. But, you know, we've also got habitats in and around Manchester that are really, really very important. The Manchester mosses, which are really, really special marshlands, areas of acid peatland around Manchester and Cheshire. And then not too far away where I'm from, where I live in North Wales, we've got some really, really important habitats. So yes, the answer is yes, absolutely. There are things to protect right here on the doorstep of Manchester. And not least of all, the waterways that flow through Manchester itself.
Great answer.
Do we have another question? Oh, plenty now. Okay. Shall we start with Giles, and then we have 2 questions at the back after.
So a question that follows up a little bit from that last question. Amanda, you've been inspiring students in Costa Rica for how many years now? A number of years.
10.
Given the university's aspiration to be carbon neutral, do you think we can justify continuing to do that? Can you make the case for why we should continue to do that?
Yes, we've had this discussion very many times.
Do we need to repeat, we repeat the question?
Oh, sorry, yeah. How can I basically justify taking, I'll shorten it, to students across the world to study biodiversity, given the university's commitment to carbon zero? Yeah? I've struggled with that question a lot, as you know. And it is a conundrum. Somebody who's a conservationist, I've worked on climate change, I know the issues around climate change. But it's almost, for me, it's ended up like a bit of a cost-benefit analysis, that, you know, the impact it has on the students and what they see and what they come away with, which is what it's all about being a teacher, I think is worth it. As I said, a lot of the students have gone on in careers in conservation, and they tell me it's because of the experience, mainly because of the experience they had in Costa Rica. They could see what you can achieve if you have a whole society that treasures their biodiversity, from the government right down to the local people, the Pura Vida. Yeah? And a lot of the students have gone on to monkey conservation, sloth sanctuaries. They go sea turtle monitoring. So they've gone on, all these students have gone on to benefit the world. So whether you think that justifies the carbon cost, I'll leave you to decide. I personally have decided it is worth it. Otherwise, I wouldn't be running these field courses still.
Can I just add something to this? Because you mentioned before that you also have the urban biodiversity study module. So perhaps a balance is also to showcase that there is a lot of work to be done home, as well as somewhere. So if we manage to balance there is also a lot of work to be done here, even on campus. We could do far more perhaps here. We may have striking the right balance, perhaps?
Yeah, I think people, a lot of these students, they go to Costa Rica, but they come back here and they put their skills and their knowledge, and use it in the UK. They don't necessarily go back and do Costa Rica. They don't necessarily go on to do. A lot of them go on. A lot of students have gone into sustainability officers. It's a stepping stone. It's like me remembering going back to University of Bath when we saw those fritillaries. It's something that we want to inspire and get that spark going in students. They can then go on and make a difference in all parts of the world, and the UK and Manchester. Going in wildlife trust officers. Some of my past students are now work for the National Trust as rangers. It's amazing what they all go on to do.
I've learned there's time for only one more question. We had 2 hands up earlier. There's 3 now. Okay, do you want to have the last word? Do you want to ask your question, please?
Yeah. Why are frogs like one of the only first species to react to climate change? And is it the same for all amphibians?
Yeah, so that's another really good question. Essentially, why are amphibians so directly impacted by, Why are amphibians impacted by climate change specifically? Well, a lot of it goes down to the biology of them. So they're ectotherms, so they regulate their temperature based off of the environment that they live in. So any subtle changes in sort of global temperature will have an impact on them. It can be really subtle on their development as a larvae and as a tadpole. If we're having water temperature increases by 2 degrees, that could have an impact on the developmental rate of their young. If we're having extreme weather events, so pronounced droughts, if we're having cyclones, these things can really damage habitats. And what the thing that happens with amphibians is we're restricting them by habitat fragmentation, by developments. You're ending up with these animals, they're in little oases almost, and that makes them more vulnerable to these weather events. If you have a huge area of protected habitat, there's more chances that some of that habitat will be sheltered from a weather event. But the frequency of these events and the regularity of these events now is causing a lot of stress on amphibian populations. So there's there's lots more reasons as well as to why. Yeah, and the canary in the coal mine, because of the fact that they're exposed to terrestrial environments, aquatic environments. They have these really permeable skins that they are really vulnerable. And yet, when we've protected them or when we afford them the space, they are adaptable and they are resilient and they can bounce back from these things. But if they are declining or vanishing, we really need to take notice. And I think from climate change is really growing in terms of the impact that it's having on amphibians. It's now the second highest cause of the threatened status sort of increases of all the amphibians we assessed. So yeah, it's a big problem.
So is we going to finish there, Maggie? Because
yes, Amanda, you're leading me all the way.
But we're all waiting for the frogs to come out.
Oh, sorry. Okay, let's get the frog out then. Yeah, let's finish with the frog out then.
Tell us what you've got, Matt.
Well, we can't have you all come here to Manchester Museum and not meet some of our amphibians. So if you do want to have an opportunity to come up and have a closer look, I have an example of our lemur leaf frogs, a critically endangered, beautiful species that Amanda talked about. And I also have Aglatinus annie, the beautiful yellow-eyed leaf frog as well, another now vulnerable species from Costa Rica. But yeah, there'll be an opportunity to speak to us in more comfortable surroundings as well after this event.
Before we rush to see those lovely frogs, can we give a last clap for Amanda, please, and Matt?